Okay, then I will start now. So are you studying? Na fakibaji? Eh?
পড়ছি ম্যাম।
পড়ছি। ফাকিবাজি হচ্ছে না না? So এই abdomen exam এ 15 of you out of 131, why you were absent in the SAQ exam? I told the batch teachers to collect the names. I don't know why you were absent. এর চেয়ে মজার ক্লাস তো আর হবে না মেডিকেল কলেজে। So you are staying at home. You don't have to come in the college in time. এর চেয়ে মজা আমরা তো এরকম পাই নাই in our lifetime. So why 15 of you were absent? I don't know how many of you I told Dr. Masoom to take the names. So you didn't do the good thing. Now I will start. So can you see?
Yes, ma'am.
Okay. So we are still in third week. I told you 3rd to 8th week is embryonic period. During 3rd week, the embryonic disc is trilaminar. That is 3-layered. From dorsal to ventral, the layers are ectoderm, mesoderm, and endoderm. Near to the endoderm is a cavity known as the yolk sac cavity. And near to the ectoderm is a cavity, the amniotic cavity. So we are still in the 3rd week. From 3rd week to 8th week, we call it embryonic period. Okay, so we will finish the 3rd week today. And in the next class, I'll just talk about 4th to 8th week. And then in the next class, I will go for your pharyngeal arches. Pharyngeal arches and pharyngeal pouches and cleft, okay? So 2 more classes. Today and another 1 day, or if I cannot finish, then 1 more class. Total 3 class and then pharyngeal arches, inshallah. So we are starting. So again, I'm talking about the events, you know it, so I will jump. So the events of the 3rd week, events are, in Bengali, we can say, ghotona, ki ghotona ghote, 3 chaptaye. So what are the events? Formation of primitive streak, formation of 3 germ layers, we call it gastrulation. Then formation of notochord from notochordal process. Then formation of cardiogenic area, then formation of allantois, then the 6th event, formation of neural tube and neural crest. And I told you this process is known as neurulation, and the process of 3 germ layer is known as gastrulation. Process of neural tube formation is known as neurulation. And this neurulation ends at 4th week. And then formation of somites. In last day, I told you the somites are the blocks of paraxial mesoderm. They first appear on each side of the embryo, just near to the notochord and neural tube on each side, and the first pair appear in the 20th day, and thereafter 3 pairs per day. And altogether, there appear 42 to 44 pairs at the end of 5th week. So mites are very important. I'll tell you in subsequent classes. And then the 8th event is formation of intra-embryonic coelom or body cavity. Today, I will start from here. And formation of intra-embryonic coelom means cavity. Coelom means cavity. So intraembryonic coelom, we know about extraembryonic coelom. We have seen that there was an extraembryonic coelom and we called it chorionic cavity. Now we will talk about the intraembryonic coelom or our body cavity. And the ninth event is formation of somatic and splanchnic mesoderm. So we have seen that the mesoderm has been divided into from medial to lateral, paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. The lateral plate mesoderm will give rise to splanchnic and somatic mesoderm. Then the 10th event is vasculogenesis and angiogenesis, that is formation of blood vessels de novo and formation of blood vessels from existing blood vessels. I said that where there is mesoderm, blood and blood vessels can form de novo, but in embryonic life and fetal life, not in us. Okay. And the last event of the 3rd week, this is not of the embryo. This is about the chorion and the villi. I will talk about the placenta later on. This is about the trophoblast, not the embryoblast. So completion of chorionic villi formation occur in 3rd week. In 13th day, there was formation of primary villi, I told you. And then in 16th day, there will be secondary villi. And in day 20, there will be tertiary villi. I will talk. Now we will start the intraembryonic coelom. Before that, I will go to intraembryonic mesoderm again. So I told it in my last class, I'm recapitulating for your betterment. So from medial to lateral, the definitive mesoderm or intraembryonic mesoderm divides into paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. So let us see the figure. So this is the cross-sectional view of trilaminar embryonic disc. And this red part is the mesoderm. This is our notochord. And here the ectoderm will form the neural tube. And the yellow cells are endoderm. Here is the yolk sac cavity. And here is the amniotic cavity. This is still trilaminar. 1 layer, 2 layer, and 3 layer. So this definitive mesoderm or intraembryonic mesoderm divides into 3 from medial to lateral, paraxial mesoderm, then intermediate mesoderm, then lateral plate mesoderm. Here also, the same thing. Now, development of intraembryonic coelom, which is the 8th event of 3rd week. Now, let us see about the coelom. So many writings are here, just listen. The primordium of intraembryonic coelom or embryonic body cavity appears as isolated coelomic spaces in the lateral plate mesoderm and the cardiogenic mesoderm. I'm showing you the figure and then I'll come here again. So what happens here? This is our lateral plate mesoderm. And if we can see from here, this is the embryo. And this one here in different color because this is taken in different book. This is red neural plate and underneath is the mesoderm. So mesoderm in the lateral plate mesoderm here and here, there appears some coelomic spaces, 1, 2, 3, and here also we can see the coelomic spaces. Look here, coelomic spaces. So the primordium of our body cavity appears as isolated coelomic spaces in lateral plate mesoderm and also in the cardiogenic mesoderm. If you remember about the cardiogenic area, where was it? In the mesoderm beyond the buccopharyngeal membrane or the precaudal plate. There is the cardiogenic area. So there also appears some coelomic spaces. Now, so these are the coelomic spaces. Now the coelomic spaces coalesce to form a single horseshoe-shaped cavity, the intraembryonic coelom or intraembryonic body cavity. So I'm showing you the figure. The coelomic spaces then coalesce. So this is the neural plate, then will form neural tube. And here lies, look at this. The coelomic spaces were here, here, here. They coalesce to form a single horseshoe-shaped cavity, single horseshoe-shaped cavity, which is the intraembryonic coelom or intraembryonic body cavity. So what happens here, I'm going back again. These isolated coelomic spaces, 1, 2, 3, 4, so many, they coalesce, এক হয়ে যায়, মিলে যায়, and they form a single horseshoe-shaped body cavity, single cavity, that is the, so spaces soon coalesce to form a single horseshoe-shaped cavity, the intraembryonic coelom or intraembryonic body cavity. So this is the intraembryonic coelom in trilaminar embryonic disc. So the embryo is still a disc. It is like a disc, big one, and this is the dorsal view. So this one is the intraembryonic body cavity. Next, the intraembryonic coelom is divided into 3 parts. I'll talk about this later on, but just remember the coelom, mute your audio. The intraembryonic coelom is divided into 3 parts. This is the, what are those? Look here, the horseshoe-shaped cavity is divided. Here will be our pericardial cavity. Here will be our peritoneal cavity or coelom. Coelom means cavity. And this part, and this part. এই জায়গাটা আর এই জায়গাটা, as it connects the pericardial cavity with the peritoneal cavity, so this is known as pericardio-peritoneal canal. Because this part of the cavity, এই জায়গাটা, this part of the cavity connects the pericardial cavity with the peritoneal cavity, so we call it pericardio-peritoneal canal. And later on, we will see that our pleural cavity form from this pericardio-peritoneal canal. So there are 3 cavities, 3 parts. So the horseshoe-shaped cavity divides into 3 parts. Etar আর দরকার নাই. Mute yourself. Now, so we have talked about the cavity, intraembryonic cavity. So intraembryonic cavity appear in the lateral plate mesoderm. Initially, there are coelomic spaces isolated, and then these spaces coalesce to form a single horseshoe-shaped cavity, and the cavity is divided into 3. This is the cavity divided into 3. Here is the pericardial cavity, here is the peritoneal cavity, and here is the pericardio-peritoneal canal. Remember the name, pericardio-peritoneal canal. Now we will go to the next event, not here. We will go to the formation of somatic and splanchnic mesoderm. This is the 9th event, and the somatic mesoderm is also known as somatopleuric mesoderm. Splanchnic mesoderm is also known as splanchno-pleuric mesoderm. Pleuri means wall here. So formation of somatic or somatopleuric and splanchnic or splanchno-pleuric mesoderm. So now, as the intraembryonic coelom appear, it divides the lateral plate mesoderm into 2 layers. As the intraembryonic coelom or cavity appear, it divides the lateral plate mesoderm into 2 layers. Let us see the figure. And the 2 layers are, okay, let us see the figure first. So look here, this is the intraembryonic coelom, এই যে সাদাটা. So it divides the lateral plate mesoderm into 2 layers. Above is the somatic mesoderm, and এই যে নিচে, this one is the splanchnic mesoderm. So as this coelomic cavity or intraembryonic coelom appear, it divides the lateral plate mesoderm into 2 layers. Above, near to the ectoderm, is the intraembryonic somatic mesoderm, and below, near to the endoderm and yolk sac cavity, is the intraembryonic splanchnic mesoderm. That is what I said, that as the intraembryonic coelom appear, it divides the lateral plate mesoderm into 2 layers. First, I will talk about the name. The names are a somatic or parietal layer or intraembryonic somatopleuric mesoderm. You can call it by three names. This is intraembryonic somatic mesoderm or intraembryonic somatopleuric mesoderm, or you can say somatic or parietal layer. And so somatic or parietal layer near to the ectoderm, and below is the splanchnic or visceral layer. We call it intraembryonic splanchnopluric mesoderm. So there are two layers, intraembryonic somatopleuric and intraembryonic splanchnopluric mesoderm. So look at the mesoderm. Next, the intraembryonic somatic mesoderm and the overlying embryonic ectoderm form the embryonic body wall or somatopleuri. I'm showing you figure, will come here. So look here, this intraembryonic, look here, look at the arrow, intraembryonic somatic or somatopleuric mesoderm, they with the overlying ectoderm. This one form the somatopleuri or the body of the embryo, body wall. And the intraembryonic splanchnopluric mesoderm here with the underlying endoderm, they form the splanchnopluric mesoderm, that is the wall of the gut tube. Just remember, wall of the gut tube. This forms the body wall and this forms the wall of the gut tube. That is what I said. The intraembryonic somatic mesoderm and the overlying embryonic ectoderm form the embryonic body wall or somatopleuri. Embryonic body wall, or soma means body. Remember, soma means body. So intraembryonic body wall or that is embryonic body wall or somatopleuri. And so look at the somatopleuri and the intraembryonic splanchnic mesoderm and underlying embryonic endoderm form the embryonic gut or wall of the embryonic gut or splanchnopluri. So this is the two wall, the body wall or somatopleuri, আর এই দুইটা মিলে হচ্ছে splanchnopluri, that is wall of the gut tube. এটার দরকার নাই. Next, again I am going to intraembryonic coelom. During the second month, that is end of embryonic life, embryonic period, that is 2 months, okay? And the intraembryonic coelom is divided into 3 body cavities. What are these? I told you earlier, pericardial cavity, pleural cavity, and peritoneal cavity. We have 3 cavity, right? Pericardial cavity, pleural cavity, and peritoneal cavity. So this intraembryonic coelom or intraembryonic cavity, which appeared in the lateral plate mesoderm, it is horseshoe shaped, and at the end of second month, they form 3 cavities: pericardial, pleural, and peritoneal. Look here, this is pericardial cavity. Here is the peritoneal cavity. Here is the pericardioperitoneal canal because it connects the pericardial cavity with the pleural peritoneal cavity. But this pericardioperitoneal canal at the end of 8th week will become the pleural cavity. So in other words, we can say that intraembryonic coelom or intraembryonic body cavity, which is horseshoe shaped, they form at the end of 2nd month or end of 8th week, the pericardial cavity or coelom, the pleural cavity or coelom. Here it will become the pleural cavity or coelom, and the peritoneal cavity. Forget about this. Now again, I will talk a few words about vasculogenesis. What is it? New blood vessel formation. Genesis means formation. So formation of new blood vessel is known as vasculogenesis. And angiogenesis means, angion has come from the Greek word angion, which means vessel. Angion means vessel, blood vessel. So formation of blood vessel from existing blood vessel by sprouting, like the branches of trees. So there is already a blood vessel, and when there is sprouting, this is called angiogenesis. Now, Just few words about this vasculogenesis and angiogenesis. It is the 10th event of third week. This is the 10th event. And what happens at the beginning of third week, vasculogenesis and angiogenesis begins in the extraembryonic mesoderm. I'm talking about extraembryonic mesoderm, not intraembryonic. Remember, of the yolk sac, of the connecting stalk, I told you. In the connecting stalk, there is extraembryonic mesoderm and also in the chorion. So let us see figure. I'll come here again. So this is the 9th or 12th day blastocyst, if you remember. So let us go to the next one. So this is a 13th day human blastocyst. This is a big cavity, the extraembryonic cavity or chorionic cavity or extraembryonic coelom. And this is our yolk sac, secondary yolk sac. Here is the amniotic cavity. Here is our germ disc. Okay. So there lies extraembryonic mesoderm. This is extraembryonic splanchnic mesoderm. Remember, extraembryonic splanchnic mesoderm. This is the connecting stalk. Also here lies extraembryonic mesoderm. And this is extraembryonic somatic mesoderm. And later on, it will form the chorion. This mesoderm, dotted. So initially, blood vessels form here in the somatic, in the splanchnic mesoderm, in the connecting stalk and also in the chorion. So that is what I said, that initially or at the beginning of third week, vasculogenesis and angiogenesis begins in the extraembryonic mesoderm of where? Of the yolk sac, extraembryonic mesoderm of the connecting stalk, and extraembryonic mesoderm of chorion. And later on, look here, another figure. So extraembryonic mesoderm, this is taken from Langman. This is the yolk sac. So this is extraembryonic splanchnic mesoderm. So blood and blood vessels form here. This is the connecting stalk. Blood and blood vessels are forming. And this is the extraembryonic somatic mesoderm or chorion. Blood vessel is forming. And later on, to Lagbena, and embryo. Now, what about the blood vessels of embryo? I was talking about blood vessels of extraembryonic mesoderm. What about the blood vessels of embryo? So embryonic blood vessels begin to develop 2 days later. Just remember after this. Embryonic first blood vessels appear in the extraembryonic splanchnic mesoderm, a connecting stalk, and in the chorion. And after 2 days, embryonic blood vessels form. Blood vessels formation in the embryo and the extraembryonic mesoderm during the third week may be summarized. So I'll not talk about this because these are a bit hard. Now, primordial cardiovascular system. Cardiovascular means, cardio means heart, blood, vascular means blood vessels, and also the blood. And I told you the whole cardiovascular system is derived from mesoderm. Remember, AJ, heart, blood vessels, and blood, এগুলো সবই mesoderm থেকে আসে, ঠিক আছে? It comes from mesoderm. Now, what happens, just remember this, that in the cardiogenic area, if you remember about the cardiogenic area, I told you that mesoderm from the primitive streak region migrate and settle beyond the precaudal plate and form an area filled up with mesoderm. We call that cardiogenic area. মানে এই জায়গাটাতে blood form হবে, heart will form. So ওই জায়গাটাতে, in the cardiogenic area, there appear 2 longitudinal endothelium line channels. We call them endothelial heart tubes. Developed during the third week and fuse to form a primordial heart tube. And the 2 tubes then fuse to form 1 single heart tube. So I will talk it later on when if I get time, I'll talk about the cardiovascular system, just primitive idea, primary idea. So paired longitudinal endothelium line channels appear in the cardiogenic area. They then fuse to form a single heart tube. The tubular heart joins the blood vessels in the embryo, connecting stalk, chorion, and yolk sac to form a primordial cardiovascular system of the embryo. So the tubular, let us see the figure. I'll come here again. This is the sagittal view. So here is the cardiogenic area, and there was two heart tube, একটা দুইটা, they fuse to form a single heart tube, and the heart tube then bends. These heart tube then joins with the blood vessels of the embryo, embryoর ভিতরে যে blood vessels গুলো হয়েছে তার সাথে, and then with the blood vessels of the connecting stalk, and then with the blood vessels of the chorion. So these heart tube, which form in the cardiogenic area from mesoderm, they form, initially there were two, দুইটা ছিল, পরে they fuse to form a single heart tube, then the heart tube bend করে. And they join the blood vessels of the embryo, intraembryonic blood vessels. Then they connect with the blood vessels of the connecting stalk, blood vessels of the yolk sac, and blood vessels of the chorion. And that is how the primordial cardiovascular system is formed in the embryo. So the tubular heart joins the blood vessels in the embryo, the blood vessels in the connecting stalk, the blood vessels in the chorion and yolk sac, and then they form a primordial cardiovascular system. So this is about the cardiovascular system. By the end of third week, the blood is circulating and the heart begins to beat on the 21st to 22nd day. So early. 21st to 22nd day, the embryonic heart start to beat. If you can remember it, otherwise, how can this embryo get its nutrition? From where? How long it will depend on mother, okay, on the yolk sac and on the mother. So the cardiovascular system is the first necessity or the first system, and the first system to work in the embryo. That is 21 থেকে 22 দিন, 21st day to 22nd day, the heart start to beat. And the cardiovascular system is the first system to reach a functional state. The system is formed, but the function yet not started. But here what happens, the system is formed and it start to function first. System form first, the functions start to form first in the 21st to 22nd day. The embryonic heartbeat can be detected ultrasonographically using Doppler technology during the 5th week. So early, about 7th week after last normal menstrual period. So 5th week আমরা কিন্তু embryonic heart tube beatingটা দেখতে পাই by the Doppler technology. So here they have done a Doppler technology, and look here, this is the embryonic disc, এই জায়গাটা পুরাটা, এখানে the amniotic cavity, and niche the chorionic cavity, yolk sac cavity, and outside a black, the huge is the chorionic cavity, kalota. And here the red portion, the heart has started to function in the 21st to 20. এই যে লালটা দেখা যাচ্ছে এটা মানে যে heart was started working. So now some clinical correlate, and বেশি বলবো না, teratogenesis associated with gastrulation, এখানে I will jump. And gastrulation means formation of 3 germ layers, right? Ectoderm, mesoderm and endoderm. So if there is any abnormalities, we call it teratogenesis. Teratogenesis means harmful effect of normal. So the beginning of 3rd week of development, when gastrulation is initiated, is a highly sensitive stage for teratogenic insult. You can understand, I talked about why the embryonic period is so much vulnerable to teratogenesis. I told you, because most of the system, every system and everything is forming during this period, 3rd to 8th week. So this is highly vulnerable period. So the beginning of the 3rd week of development, when gastrulation is initiated, is highly sensitive stage of teratogenic insult, not the beginning of 3rd week to 8th week, the whole embryonic period. Now, high doses, just go on hearing, don't write anything. High doses of alcohol at this stage kill cells in the anterior midline of the germ disc, producing a deficiency of the midline in craniofacial structure. We call it holoprosencephaly. এটা দরকার নাই. Next we go. দরকার নাই. And gastrulation itself may be disrupted by genetic abnormalities and toxic insult. So insult can be X-rays. Insult can be any drug or any toxin, or it can be genetic. And there is a term known as caudal dysgenesis or cynomyelia. What happens here? Insufficient mesoderm is formed in the caudalmost region of the embryo. So in the caudal part, there is insufficient formation of mesoderm. Mesoderm na hole hat pa hobe na, onek kichui hobe na. So there is insufficient formation of mesoderm in the caudal part of the embryo. Jerokom ekhane ki hobe, because the caudal mesoderm contributes to formation of lower limbs, urogenital system, lumbosacral vertebrae. So abnormality hobe ekhane, and the child, এটা দরকার নাই, just child is একটা, I want to show you a figure. So this is caudal dysgenesis. The limbs are fused here. So this is the cynomyelia. আর এখানে we call it, আর এমনি normally we call it mermaid baby. So this is actually caudal dysgenesis. That is insufficient formation of mesoderm in the caudal region of the embryo. So এটার আর দরকার নাই. And next we will go to our last event, that is the development or further development of core unit villi. Why I have written further development? Because in the 13th day money 2 week na, 2 week. So the primary core unit villi was formed in day 13. That is end of second week. But we are going through the 3rd week, not in the 2nd week. That is why I have written further development of core unit villi, that is trophoblast, 11th events of 3rd week. Now what happens? So I have told you about the primary core unit villi again. So primary core unit villi, look here. So this is our primary core unit villi. I told you that there is proliferation of cytotrophoblast here, AJ cell gula, and it is surrounded outside by syncytiotrophoblasts, bottle green in color. I told you there is finger-like projection. First of all, multiplication of cytotrophoblast, অনেকগুলো cell হবে. Then they will form finger-like projection. এই যে একটা, দুইটা, তিনটা, চারটা দেখাচ্ছি, and they are surrounded by syncytiotrophoblasts. So we call it a primary villi, and this primary villi is formed in the 13th day. That is the end of 2nd week. Next, so now we have to see the primary core unit villi then consists of a cytotrophoblastic core covered by syncytial layer. So আমরা কি বলতে পারি যে there is a cytotrophoblastic core here, এখানে অনেকগুলো cell and surrounded by syncytiotrophoblasts. So we can say that the primary core unit villi consists of a cytotrophoblastic core covered by a syncytial layer. If I do a cross section, cut section here, এখানে যদি আমরা এরকম করে একটা cut section করি, like here, এখানে এরকম ভাবে, then what will we see? We will see this. So inside is the cytotrophoblastic core and outside is the syncytiotrophoblasts. This comes in the exam, in the final exam MCQ question. Describe the development of placenta. So you have to know it. So this is the syncytiotrophoblastic, inner is the cytotrophoblast. So this is a primary villus formed during the 13th day. Next, early in the 3rd week, that is 16th day. So we have come to the 3rd week now. Extraembryonic mesoderm, somatopleuric mesoderm, grows into the primary villi, forming a core of mesenchymal tissue. Let us see the figure here. So what happens? That extraembryonic somatic mesoderm, look here, এগুলো সব extraembryonic somatic mesoderm, they grow inside, ভিতরে ঢুকে যাচ্ছে, they grow inside the primary villi, এই যে এখানে একটু ঢুকেছে, এখানে, and now we call it a secondary villus. তাহলে কি হলো? What happens? Early in the 3rd week, extraembryonic somatopleuric mesoderm, grows into the primary villi, forming a core of mesenchymal tissue, that is a core of mesodermal tissue, একই কথা. So we call it a তাহলে আমরা এটাকে বলছি, we call it the villi at this stage is known as a secondary villi which cover the entire surface of the chorionic sac. আমরা এটাকে বলছি secondary villi. So let us see the secondary villi. This is the primary villi and this is the secondary villi. The mesoderm or mesenchymal core is here. Outside is the cytotrophoblast and একদম বাইরে হচ্ছে syncytiotrophoblast and it develops during 16th day, secondary villi. Now come to the tertiary villi. By the end of third week, that is day 20, mesodermal cells in the core of the villi begin to differentiate into capillaries and blood cells. So I told you where there is mesoderm, there is the development of blood and blood vessels, that is capillaries and other vessels. So by the end of third week, that is 20th day, mesodermal cells in the core of the villi begin to differentiate into capillaries and blood cells. তাহলে so look here, এখানে একটু এই জায়গায় এগুলা ব্লাড সেল ফর্ম হয়ে গেছে। সো এটা না, আমরা এখানে যদি একটু দেখি, এটা না, so here this mesenchymal tissue এখানে আছে, এখানে blood cells and blood vessel will form and then we will call it a tertiary villi. So we have talked about the primary villi, the secondary villi and the tertiary villi. So let us see now the villi are called tertiary chorionic villi or definitive villi. Tertiary chorionic villi or definitive villi. Actor অনেকগুলা নাম. So this is the three sort of villi. 13-day primary villi, core of cytotrophoblast surrounded by syncytiotrophoblast. 16-day mesenchymal tissue or mesoderm grow here. So, and বাইরে cytotrophoblast and then syncytiotrophoblast. And during the day 20, blood vessels and blood form here, capillaries form here in mesoderm. So this is the tertiary villi. So we have finished it now. Meanwhile, now what happens? The cytotrophoblastic shell outside, একটু আমরা go back. This cytotrophoblastic shell, remember the cytotrophoblastic shell outside, cytotrophoblastic cells in the, cells in the tertiary villi penetrate progressively into the overlying syncytium until they reach the maternal endometrium. I'm showing you a figure, will come here again. What happens, this cytotrophoblastic cell, এখানে, they just erode the syncytiotrophoblast. Erode করে, they will come out. এটা বেরোয় আসে. So the here, look here, এখানে যে cytotrophoblastic cells, they erode the syncytium overlying. এখানেও সব জায়গায়. And they then come out is cytotrophoblast. That is what I said, that the now এটা না, sorry. Cytotrophoblastic cells in the tertiary villi penetrate progressively into the overlying syncytiotrophoblast until they reach the maternal endometrium. That means they just penetrate and until they reach the maternal endometrium. And now they establish contact with similar extensions of neighboring villi, that is অন্য জায়গার cytotrophoblastic cell এসে এর সাথে connect করে, forming a thin outer cytotrophoblastic shell. আমরা এই ছবিটা দেখি. Let us see here. We have taken a portion of this villas, tertiary villas. Look here. What should it have been? এটা হওয়া উচিত ছিল এরকম, right? যে ভিতরে cytotrophoblast and outside the syncytiotrophoblast. What happens? The cytotrophoblast here, they penetrated and come out to establish contact with the endometrium of the mother. And then they connect with the similar extension of the neighboring villas. এখানেও একই. Next, আরেকটাও একই, same. And they ultimately form a cytotrophoblastic shell. Outer cytotrophoblastic shell. Initially, we have seen that the cytotrophoblast lies inside and the syncytiotrophoblast outside. But at one time, at the 20th day, when the tertiary villus has been formed, the cytotrophoblast penetrate the syncytiotrophoblast. এখানেও penetrate করেছে, এখানেও. And they come in contact with the neighboring cytotrophoblast এখানে, and they form a cytotrophoblastic shell. Now, what has happened? This area, the red area, এই জায়গাটা অথবা এই জায়গাটা, this is known as, this is a villus now, এটা একটা villus, এটা একটা villus, this is known as the intervillus space. এটা দেখেছো, এটা intervillus. Now কি হলো, intervillus space is lined by syncytial trophoblast, তাই না? Intervillus space is lined by syncytial trophoblast and it is filled up with maternal blood. এই লালগুলা, the red color, this is all maternal blood here, here also, and they have come from maternal blood vessels. Look here, maternal vessels in the endometrium, because these cells has eroded maternal blood vessels. I told you, খেয়ে ফেলেছে maternal blood vessels, so maternal blood seeps here, হ্যাঁ, আস্তে আস্তে করে এখানে ঢুকে পড়ে. So this is the intervillus space. So I told you, if you remember, for a second week, the 9th day, the lacunar stage of development, and lacunae were nothing but the primordium of intervillus spaces, বলেছিলাম. So this is the intervillus. So what happens? Again, আমি একটু বলি. They establish contact with the similar extension of the neighboring villi, forming a thin outer cytotrophoblastic shell. So look at the shell, and এটা দরকার নাই. The cytotrophoblast shell gradually surrounds the cornea and attaches itself to the endometrium. I have told you. And outside there, we will get the cytotrophoblastic shell and will connect the whole thing, the cytotrophoblastic shell, with the maternal endometrium. Now, capillaries in the tertiary villi make contact with capillaries developing in extraembryonic mesoderm of chorionic plate and connecting stalk. I already told you that these capillaries in the chorionic villi, they make contact with the blood vessels formed in the chorionic plate, extraembryonic mesoderm of, because here lie extraembryonic somatic mesoderm and here lie extraembryonic somatic mesoderm. This is the connecting stalk. So these capillary of the tertiary chorionic villi, they make contact with the capillaries of the chorionic plate and with the capillaries or blood vessels of the connecting stalk. So that is what capillaries in the tertiary villi make contact with the capillaries developing in the extraembryonic mesoderm of the chorionic plate and in the connecting stalk. তাহলে এটা হয়ে গেল. And next we can see capillaries of the chorionic plate and the connecting stalk then in turn establish contact with the intraembryonic circulatory system connecting the placenta and the embryo. And later on what will happen, these capillary, এই যে capillary গুলো formation, এটা ভুলে যাই আমরা, we will go to that. These capillary then will make contact with the blood vessels of the embryo, পরের দিকে embryo, and these capillaries will make contact with the, so these capillaries of the tertiary chorionic villi, they make contact with the blood vessels of the chorion, that in the chorion there is extraembryonic mesoderm. তারপরে এরা make contact with the blood vessels of the connecting stalk এর মধ্যে extraembryonic mesoderm, and then they contact with the intraembryonic blood vessels. So thus they establish the maternal and embryo, that is they, না এটা না, sorry. So they establish contact with the intraembryonic circulatory system connecting the placenta and the embryo. Placenta we will talk later on, আমরা আরো পরে বলবো. So we have finished, and hence by the end of 3rd week, when the heart begins to beat, embryonic blood start to flow slowly through the capillaries. দরকার নাই. Now কি হবে, oxygen and nutrients in the maternal blood in the intervillus space diffuse to the walls of the villi and enter the embryonic blood. Let us see the figure, I'll tell you. Now these capillaries era make contact with the blood vessels of the connecting stalk, chorion, and then embryo. Now what happens, look here, blood here is maternal blood, ekhane blood, maternal blood, right, rich in oxygen and nutrients, and ekhane blood, the embryonic blood in the capillaries, they are not rich in nutrients and not rich in oxygen, rich in carbon dioxide. So here there will be diffusion. এই যে এইভাবে, through this there is diffusion. So oxygen from here, nutrients from here will come into the capillary here in the placenta, in the chorionic villi, and carbon dioxide and waste from here will go in this direction to the villus spaces. তাহলে what we can see, diffusion of oxygen and carbon dioxide nutrients will occur through this barrier, this membrane. So this is a blood এই জায়গাটাকে আমরা বলি, we call it a blood barrier. So in the embryonic life or fetal life, oxygen and nutrients will have to traverse through there is a barrier, we call it blood barrier, because blood here, maternal blood here, capillary, the fetal blood or the embryonic blood, so blood barrier. But in our life, in adult life, there is a blood gas barrier, right? Blood in the capillary of the pulmonary capillary, and then in the alveoli there is gas. So the fetal lung is not working, fetus is embryo or fetus is not breathing, so it is taking its oxygen and nutrients and everything through maternal blood. So there is a blood barrier. That is what I have said, that oxygen and nutrients in the maternal blood, in the intervelar space, diffuse to the walls of the villi and enter the embryonic blood vessels or blood. Carbon dioxide goes in opposite direction. So now there are some types of villas, sometimes comes in the exam, আমি একটু বলে যাই, stem villi or anchoring villi, and branch villi or free villi or terminal villi. Just I'll tell you what is a stem villi or anchoring villi. Okay. So this villus, এই পুরা villusটা হচ্ছে এখান থেকে, it start from here. This is a stem villi or anchoring villi. So this is the main villus, and it anchor the whole thing with the maternal endometrium. So this is the anchoring villi. ঠিক আছে? আর পাশে যেগুলা ছোট ছোট branch villi, এই যে here is a branch villi, here is a branch villi, here is a branch villi. So the villas are two types, the stem villi or anchoring villi, branch villi or free villi or terminal villi. একটা তিনটা না. One has three names, branch villi or free villi. Now you can understand that through this branch villi or free villi or terminal villi, gaseous exchange occur, not with the stem villi. The function of the stem villi is to anchor. The function of the stem villi is to anchor. But this free villus, through this there is diffusion. এদিকে. So through the stem villi there is anchoring, and to the branch villi or free villi or terminal villi there is exchange. So villi that attach to the maternal tissues through cytotrophoblastic shell are called stem or anchoring villi. Maternal tissue with the cytotrophoblastic shell is the stem villi or anchoring villi, and the villi that grow from the sides of the stem villi are terminal villi or free villi or branch villi. So villi that grow from the sides, side থেকে যেটা grow করে, that is the stem villi or terminal villi or branch villi. So it is to the walls of the free or branch villi that the main exchange of material between the blood of the mother and the embryo takes place. So you can understand, নিজেদের বুদ্ধি থেকেই বুঝবো আমরা, that it is to the wall of this branch villi, এগুলো দিয়েই mainly gaseous exchange of nutrients and everything occur from the embryo or fetus to the mother, and from the mother to the embryo or fetus, right? So we have finished now. So next, so we have finished this one also. Some clinical correlate একটুখানি বেশি না. Now what happens? Abnormal growth of trophoblast. I'm talking about that. My last event was development of chorionic villi. So sometimes there can be, what happens, embryo may die and the chorionic villi do not complete their development. That is, they will halt in some stage of their development. চুপ হয়ে থেমে গেল. That is, they do not become vascularized to form tertiary villi. যদি vascularized না হয়, then that is not a tertiary villi, right? So কি হবে? We call it, এটাকে আমরা, these degenerating villi form cystic swelling. That is, they don't become tertiary villi because the embryo has died. Embryoটা মরে গেছে, তো tertiary villi formation হলো না, there is no formation of tertiary villi. What will happen to the secondary villi? They will swell, তার ভিতরে maybe water or something like that, they form cyst. Cyst is something, a hollow structure inside gas or liquid. So cystic swelling, we call it hydatidiform mole. Hydatidiform mole, মোলের মতো উঁচু উঁচু, which resembles a bunch of grapes. মনে হয় কি খালি বেরোলে যে একটা আঙ্গুর, bunch of grapes. Hydatidiform mole resembles a bunch of grapes. And the moles exhibit variable degrees of trophoblastic proliferation and produce excessive amount of HCG. A pregnant lady, what is a pregnancy test? What we do, we take the urine or blood to confirm pregnancy. If we see that there are certain levels, if we see there is high level of HCG, human chorionic gonadotrophin, which is secreted by the syncytial trophoblast, I told you, তাহলে আমরা we can say she is pregnant. But there is a level, if it is high enough, then we can say, and we will have to correlate with that other function also, then we can say that there is hydatidiform mole. And and also 3 to 5% of the hydatidiform mole develop into malignant trophoblastic lesion. It can become, hydatidiform mole is benign, not malignant, not bad, but 3 to 5% of the hydatidiform mole can become malignant. That is, it becomes bad. তখন আমরা, we call it choriocarcinomas. That is cancer. And if there is choriocarcinoma, the HCG level is much more high. So we have finished, and some more develop after responded, so we finish it here. Choriocarcinoma এখন দরকার নাই. So summary of the third week. So major changes occur in the embryo as the bilaminar embryonic disc is converted into a trilaminar embryonic disc during gastrulation. So third week, the gist of the third week is that major changes occur here. The bilaminar embryonic disc, that is the epiblast and the hypoblast, they are converted into trilaminar embryonic disc by the process known as gastrulation. And the events, আমি এখানে shortly বলে যাচ্ছি যে ওখানে কি হবে, there will be formation of primitive streak, তারপর gastrulation, formation of cardiogenic area, notochord formation from notochordal process. So this is so important. Neural tube formation, we call it neurulation, neural crest formation, and we have seen the structures from neural crest. Somoid formation, very important during day 20 from the paraxial mesoderm. And intraembryonic coelom has formed in the lateral plate mesoderm. Initially, there were coelomic spaces. The coelomic spaces coalesce to form a single horseshoe-shaped cavity, and the cavity divides into 3 parts. The pericardial cavity, the pericardio-peritoneal canal, which later on become the pleural cavity, and the peritoneal cavity. Formation of blood vessels and blood, completion of chorionic villi formation. So these are the events of third week, and to summarize, we can say this. So if you remember the events, we can easily remember. So this is the end of third week, which is very important. So many questions comes here in the exam. There is just interesting a thing that a female infant was born with a large tumor situated between her rectum and sacrum. A diagnosis of sacrococcygeal teratoma was made, and the mass was surgically removed. I told you that sacrococcygeal, whenever the primitive streak, there is remnant of primitive streak, there will become a tumor, and we call it sacrococcygeal teratoma, and it can be surgically removed. So what is the probable embryological origin of this tumor? আমি যদি জিজ্ঞেস করি যে কেন এটা হলো, what is the probable embryological origin? So answer should be, or we can arekta question korte pari, explain why these tumors often contain various types of tissues derived from all 3 germ layers, because they have come from epiblast, and we know that epiblast is the source of all the 3 germ layers. Answer should be like this. Does an infant sex make him or her more susceptible to the development of one of these tumors? Yes, female are more affected. Answer, sacrococcygeal teratomas arise from remnants of primitive streak. Because cells from the primitive streak are pluripotent, I told you. That is, they're partially differentiated cells, right? Pluripotent epiblast. The tumors contain various types of tissues derived from all the three germ layers. Kiki germ layer from dorsal to ventral, ectoderm, mesoderm, and endoderm. And there is a clear-cut difference in the incidence of these tumors with regard to sex. They are 3 to 4 times more frequent in girls than in boys, but we don't know why. But there is a clear-cut difference. After research or after observation, scientists have found that the females are more vulnerable, but we don't know why females are more vulnerable, more affected than male. দেখা গেছে 3টা মেয়ে 4টা মেয়ের হলো একটা ছেলের। হ্যাঁ, so why this happens we don't know, but it happens. So this is the end of today's class. Next, we will discuss about 4 to 8 week, and we will complete the embryonic period. And then, inshallah, we will go for the pharyngeal arches, that is pharyngeal apparatus. And my advice to you all is to read. Don't stop your study. Time flies, time goes on. Maybe due to this situation, the pandemic situation, somewhat we are not able to give you the education in proper way.